If you remember the very beginning of this course, I talked about how we are always assaulted by viruses every hour, and we're healthy most of the time because we have a great immune system. So today we're going to finish up a little discussion of that immune system. We're going to talk about adaptive defenses. Now, last time you may remember we talked about intrinsic defenses, the ones always present in an uninfected cell. We talked about a few examples of those: apoptosis, autophagy, RNA silencing, antiviral proteins. We talked about innate immunity, which is induced by infection. But today we're going to talk about the adaptive immune system, which is tailored to the pathogen. None of the other two defenses are tailored. They are general, nonspecific defenses meant to inhibit all viruses. But if things go badly, if the intrinsic or innate defenses can't handle the infection and it goes out of control, adaptive immune system comes to play. And not only is it tailored to the pathogen, but it gives us memory so that if we should encounter the pathogen again, we'll be able to respond more quickly. Last time we ended up at this point where we have a monolayer mucosal surface of epithelial cells. It's infected with a virus. The cells that are infected can sense that the virus is present. They produce cytokines. They may also produce apoptotic bodies. The dendritic cells, macrophages as well, come into this area. They're attracted by the cytokines produced by the infected cell. They come into the area. They may produce more cytokines as well to try and eliminate infection. And they will also sample the apoptotic bodies that are being produced from the cells, or maybe these cells are producing virus particles. These dendritic cells will sample them. And they take those and go to the lymph node. Remember, the dendritic cells get activated, so they grow these long processes. They go to the lymph node, and then they present the proteins that they've acquired at the mucosal surface, and they say, is this foreign? And then if it is foreign, we initiate an adaptive response. So someone asked last time, what is the threshold at which point we go to a full-blown adaptive response? It's not an easy question to answer, but presumably, let's say a dendritic cell came in here and made a lot of interferon, picked up some proteins, viral proteins, went back to the lymph node. In the meantime, the infection's over, right? Let's just say it's cured by virtue of the cytokines produced. The dendritic cell will go to the lymph node. It might instruct a few T cells to start proliferating, but it won't continue to get cytokines. That dendritic cell may go away and no others may come in. So the T cells will proliferate to a certain extent, but they really require continuous cytokines in order to proliferate, which means you have to have a continuous infection. So if you get above a certain threshold of infection and there are lots of DCs going in and feeding cytokines to the lymph node, then you will get activation of the adaptive response. And if you don't, it will subside and then just rest for next time. So today we're going to talk about this step, when the dendritic cells go in the lymph node and instruct the adaptive response. And of course, the end result is going to be the production of antibodies specific for the virus and cytotoxic T lymphocytes, both of which are meant to control virus infections. So when the dendritic cell goes in, here's what it does. Here's a dendritic cell on the upper left. It's yellow, and it's been activated by picking up proteins from the infected cell. It's now in the lymph node, and it will present what it's got to either a CD8 T cell or a CD4 T cell. These are the two main types of effector cells in the lymph node. And the dendritic cell is going to present the viral antigens on its surface to these two different types of cell. And the way that happens is somewhat complex and different for CD8 and CD4s. So remember, the DC has picked up apoptotic bodies or pieces of viruses. Those viral proteins are then processed and sent to the cell surface. We'll look at how that happens in a moment. And they're shown here as red lines labeled viral peptides. So these are very short, 8 to 10 amino acid long versions of the original viral protein. And for CD8 cells, they are presented to the CD8 cell in a molecule called MHC1, major histocompatibility protein type 1. So it's a cell surface protein, and it carries the peptide. You could look at it as a hot dog in a bun, presenting this peptide to the CD8 cell. And then the CD8 cell, we have lots of different CD8 cells in us, which are all directed against foreign proteins. All the ones directed against us have been eliminated during our development. So the T cell, if it responds to this peptide, it knows it's foreign and it will begin to proliferate. Now, in addition to having the MHC1 present the peptide to the T cell receptor, which is on the T cell requires, in order for it to become activated, it requires interaction with a cell surface protein on the dendritic cell. CD80, CD86 interacts with a receptor on the T cell. And it also requires cytokines produced from the dendritic cell. So 3 signals are needed in order to activate the T cell. The protein, the peptide presented in the MHC1, the other ligand receptor interaction, and cytokines. And so if these cytokines aren't continuously produced by additional, say, dendritic cells coming in here, eventually the T cells won't proliferate any longer. So that's the way the dendritic cells present the viral peptides to CD8 cells, and these will eventually become activated, they will proliferate, and they will become cytotoxic T cells. And these will end up killing virus-infected cells. They'll come out of the lymph node, of course, and circulate, go to the area of infection, and try and kill the virus-infected cell. We'll have more to say about that in a moment. The dendritic cells also present these viral peptides to CD4 positive T cells, again in the lymph node. They do so via a different MHC, the MHC2 in this case. Here it's presenting a viral peptide. Again, the T cell receptor on the CD4 positive T cell is recognizing the peptide in the MHC2. There's also a second set of receptor ligand interactions that occurs, and these cells also require cytokines produced from the dendritic cell. Remember, the cytokines are produced in response to sensing something foreign, like a viral protein or a viral RNA. Now, an easy way to remember which is MHC2. You multiply it times 2, and you get 4. And that's the linkage between MHC2 and CD4 cells. And MHC1 is for CD8s. All right, so then the CD4 cell, upon recognizing this viral peptide as foreign, will also become activated and proliferate, and it will become a helper T cell, what we call a helper cell, which will then make cytokines that are used either in the development of antibody or in the maturation of the cytotoxic T cell. And we'll come back to that later. So this is what the dendritic cell does in the lymph node. It's very different from NK cell recognition, which we talked about yesterday, Monday, which does not involve finding viral antigen on a cell surface. Now this is looking at the antigens that the DCs are presenting and asking if they're foreign or not. So in us we have many, many different T cells, each of which recognizes a different peptide. And they're all foreign, because as I said previously, the ones that recognize us are eliminated during our neonatal development. So among all those T cells, there is at least one that's going to recognize a foreign peptide. It's really remarkable, but any viral or bacterial or any foreign antigen we put in there is going to be sensed by a T cell, and then they will proliferate. Let's talk a little bit about the antigen presentation. And we talked about MHC1 and MHC2. Here we're looking at class 2 presentation. This largely happens in antigen-presenting cells like dendritic cells. So this is what's happening in the dendritic cell. The dendritic cell have taken up proteins from the environment, and they may or may not be viral. They're always sampling proteins. And these proteins are processed through the endocytic pathway. So these proteins are degraded to peptides in the endocytic pathway. Meanwhile, the MHC class 2 molecule is synthesized in the ER. It's shipped through the Golgi, through vesicles, and then those vesicles containing the MHC fuse with the incoming endosomes with the viral peptides, and the viral peptides are loaded onto the MHC2 molecules. Those are those orange rectangles there. Again, very short peptides, and then those go to the surface, and now these vesicles transport the loaded class 2 to the surface, vesicle fuses. Now we have class 2 at the surface of a dendritic cell, and it can present it to the CD4 T cell. Remember, the class 2 is going to present to a CD4 T cell. And if that's foreign, then the T cell will become activated. All right, so that's the process. This is called the exogenous antigen presentation pathway, because these DCs are taking up material from the outside of the cell, sampling it. And of course, if this is foreign peptide, the T cell is activated. If it's not foreign, there's no activation, and the T cell hangs around and waits for another dendritic cell to interact with it. This is the same sort of scheme for MHC class 1. And we call this the endogenous antigen presentation pathway. Here, we're looking at basically virus-infected cells, which are producing proteins in the cell. And here's a viral protein, this squiggly orange thing on the left here. And in this case, the peptides are derived from the protein inside the cell by the proteasome, a very large protein processing apparatus that's found in the cytoplasm of the cell. So in any virus-infected cell, this could be a dendritic cell if it were infected, but mostly it's all of our other cells that are infected in our body. The proteasome is making peptides. At the same time, these cells are making MHC class 1 molecules. Very similar to class 2, they will present the peptide in a cleft on their surface, but MHC class 1 are on most of our cells, as opposed to 2, which is mainly on antigen-presenting cells. So MHC class 1 is produced, and it is loaded with the peptides. The peptides are actually shipped through the ER membrane in this transporter protein here, which is called the TAP protein, or the transporter associated with antigen processing. So this is an active pump that moves the peptides through so that then they can join up with the class 1 molecule in the lumen of the ER. And then these class 1 molecules bearing the peptides are shipped out through the Golgi apparatus, just like any secreted protein, through vesicles. They reach the surface, and now we have MHC1 with a peptide. And if this is a virus-infected cell, this could be a CD8 cell, which is recognizing it. Of course, this could also be a dendritic cell that's asking the CD8 if it's foreign or not, but in many cases, this is actually a cytotoxic T lymphocyte that's going to kill this cell. We'll see how that works a bit later. Now, in the previous slide, there was something missing which didn't show up. I meant to tell you. So at every step of the immune response, there's viral antagonism. Class 2 presentation, we don't know a lot of antagonisms. The one main one, which failed to make it on this slide, is that transcription of the MHC2 mRNA is actually inhibited by a herpes virus, cytomegalovirus. That's really the only one we know about to inhibit this whole pathway. Class 1 presentation, on the other hand, is inhibited at many stages by many viral proteins. So for example, these circles are showing that the actual synthesis of class 1 mRNA or protein can be inhibited by viral proteins. The movement of the class 1 through the ER can be inhibited. The transporter itself is the target for inhibitory viral proteins. The proteasome is the target of an inhibitor as well. So lots of viral inhibitors of class 1 function. On the bottom here are 3 herpes viruses that inhibit the TAP complex in different ways. This is herpes simplex virus, which makes a protein which is called ICP47. It inhibits peptide binding to the transporter. So the transporter can't be shipped through to be loaded onto the class 1 molecule. Here is a human cytomegalovirus inhibitor, US6, which blocks ATP binding. So the transport of peptides through the TAP transporter requires ATP, and this viral protein blocks that, so it can't be transported. And then Epstein-Barr virus protein binds both ATP and inhibits peptide binding. So these, you can imagine, if you inhibit loading of the peptide onto class 1, that infected cell is not going to be sensed as foreign. It won't be lysed, so it will survive. And in fact, all these herpes viruses, as we will see in a couple of weeks, cause persistent infections. They're not cleared. And the reason they're not cleared is in part because of these inhibitory activities. And cytomegalovirus in particular has so many different antagonistic activities at the level of MHC function. It's really quite amazing, and that's why we all have cytomegalovirus in us. Each and every one of you has this virus, but you're fine. So here are some listings of the antagonisms of MHC1. So here is inhibition of class 1 synthesis. There's a lentivirus protein that can do that. The TAP processing transporter machine is inhibited. Its synthesis or its function can be inhibited by different viral proteins. You can see all the herpes viral proteins that are involved here. So that's one step. Then the transport of the class 1 protein can be inhibited. Some viral proteins, so on the right are the viral proteins that do the work. Some viral proteins make the class 1 stay in the ER. They never make it to the cell surface. Some of them retain it in the pre-Golgi. Some of them direct it to the cytoplasm where they can't get to the cell surface. Some of them dislocate it to lysosomes where they're degraded. And then there are viral proteins that actually bind to cell surface class 1 molecules and prevent them from being recognized by the CD8 cells. And then there's some viral proteins that increase endocytosis of class 1. So the class 1 gets to the surface, but it's immediately endocytosed, so it can't be seen by the CD8. All right, so these are all antagonisms that these viruses have to do, otherwise they're eliminated. Right? And so they have also been useful for understanding how a lot of these steps in antigen presentation works. So we're back to the dendritic cell in the lymph node. It's presented some foreign peptide to T cells, and it's recognized as foreign. What happens? Well, the cells begin to proliferate. Only about 1 in 10,000 or 1 in 100,000 of your B and T cells will recognize any particular antigen. So we don't have a lot of them. There are millions of T and B cells circulating in us, but just a few recognizing each antigen. But within a week or two after a dendritic cell going into a lymph node and presenting a foreign peptide, those cells will multiply many, many times. They're amplified from 1,000 to 50,000 times. So that one T cell that recognizes the peptide just divides like crazy and make lots of clonal derivatives of the original T cell, which is now recognizing that specific viral peptide. So that's the key here. They are specific for that viral peptide. And this is why when you get certain illnesses, you get swollen lymph nodes. That's what lymphadenopathy means. So if you get a cold, typically, you can feel swollen lymph nodes under your throat. And that's a sign that you are making an immune response. Next time it happens, just think about it. Wow, my dendritic cells have gone in there and my T cells are proliferating. That's what's going on. That happens in other places of your body, but you typically can't feel it. So if you have a GI infected, your mesenteric lymph nodes are also swollen, but you can't feel them because they're deep in your gut cavity. All right, so that's what's happening. You get lots of proliferation, and those cells will then move out into your circulation and home into the infected area and do their thing. This first question is, antigen presentation by dendritic cells in the lymph node has which features? 1, presentation of viral peptides on MHC1 to CD8 cells. 2, presentation of viral peptides on MHC2 to CD4 cells. 3, viruses counter MHC2 at the transcriptional level. 4, viruses counter MHC1 at many levels, such as peptide transport. 5, all of the above. 86% of you answered E, all of the above, and it's correct. They're all right. So those of you who answered individual ones, you're right on the individual answers, but it's all of them. Those are all characteristics of antigen presentation by DCs. All right, so now let's talk a little bit about these cells that get activated and proliferate in the lymph node. What is it that they do? And that involves talking about the effectors of the adaptive response. And by that we mean the antibodies, the cytokines, the cytotoxic T lymphocytes. So here we have a variety of antigens which are sensed by, in this case, dendritic cells, as we've discussed, presenting antigens in the lymph node. And you can have, of course, those present antigen to 2 different kinds of cells, to CD4 or CD8 cells in the lymph node. And the top part here is just tracing the development of these cells through the bone marrow and the thymus. But what we're concerned about here is in the lymph node. So when the CD4 cell recognizes a foreign peptide presented by a dendritic cell, it responds by proliferating. It also makes a lot of cytokines. So that's the main function of these CD4 cells. They're also called T helper or TH cells. And they make cytokines that lead to the differentiation of either CD8 cells or to the production of antibodies. And those are called Th1 cytokines or Th2 cytokines. Th1 cytokines help CD8 cells develop. Th2 cytokines help B cells that are making antibodies to develop. And we'll talk about what determines that in a bit. When the antigen is recognized as foreign by a CD8 cell, the CD8 cell proliferates and matures into a cytotoxic T lymphocyte, which will then go out into the circulation and find infected cells and lyse them. And we'll talk about how that happens. So the CTL, cytotoxic T lymphocyte, is one of the big effectors of the adaptive response. On the other hand, many viruses induce the production of antibodies. So the antigen is recognized as foreign by B cells in the lymph node. And B cells have on their surface a membrane-bound antibody molecule of one type, which is specific for a particular epitope. And if it matches up with the antigen presented by the dendritic cell, these B cells will proliferate. They will become what we call plasma cells, which are made to secrete antibodies. These cells can make thousands of antibody molecules per second. They are machines dedicated to making antibodies. So the antibodies are the little red Y molecules that will combine with viruses, and we'll talk in a moment about how they block infection. So these plasma cells go out of the lymph node and go into your bloodstream and other areas, so you end up having antibodies in the blood. There are also antibodies that are produced that will cross epithelial barriers, so they end up on your mucosal surfaces, your respiratory tract, your gut, etc. And they will be trying to control the virus infection. All right, so that's what the output is of this DC interaction. Cytotoxic T lymphocytes, antibodies, and the CD4 is making cytokines that regulate these two processes. So let's talk about antibodies and how they work. The antibodies that block virus infection, we call these neutralizing antibodies because they neutralize infectivity. Yes?
In the previous slide, it seemed that the CD4 cells produce both forms of both forms of cytokines.
Right.
You seem to say that the CD8 cells may respond. The CD8 cells respond to Th1.
Right.
But the CD4 cells produce both.
So CD4s produce a mix, sometimes more of one than the other. We'll talk about that and how that happens in a moment. But the Th1 cytokines help CD8 cells differentiate into CTLs, and the Th2 cytokines help the B cells differentiate into plasma cells. So yes, CD4s can make both, depending on what kind of stimulus they get from the dendritic cell. So antibodies, of course, are Y-shaped molecules. You probably all know this, made up of heavy and light chains. And at the tip is a variable site which combines with the antigen. We make many different kinds of antibodies of different specificities. And these are essential defenses against many virus infections. When we immunize ourselves with vaccines, we are trying to produce antibodies that will block virus infection. So these antibodies, as I said, are present in the blood, and they're present at mucosal surfaces. They combine with virus particles, and they prevent them from infecting. So you can see if you have enough antibody around, eventually you will prevent virus infection from proceeding. So again, they can be in the blood, and if a virus has a viremic stage, they will bind to that virus. There's also a form of antibody called IgA, which is secreted and is present at your mucosal surfaces, your respiratory tract, urogenital, GI tract, etc. So these can block entry of the virus at an early stage. Now, most of the time we think that antibodies prevent infection. So I'll show you a time course in a moment. By the time you make antibodies after a virus infection, it takes about 2 weeks to do this. In most infections, the infection is winding down already. So the antibodies don't help you all that much, although there are a few virus infections, as I've shown here, where we know that antibodies are important for recovering. Mostly we think that cell-mediated immunity, cytotoxic T lymphocytes, are what cure us from the virus infection. However, the next time you get infected, you will now have a very rapid memory antibody response. You'll make antibodies very quickly, and they'll probably help prevent infection. And of course, that's how vaccines work as well. Yeah.
Are IgAs always present then on the mucosal surface?
So the question is, are IgAs always present? So they are largely present for mucosal pathogens that infect that mucosal surfaces, and the cells that produce the IgA are just beneath, and they'd be stimulated to produce them. But a virus that does not reach a mucosal surface may not make IgA. Yeah. So here's an example of a neutralization assay. We take antibodies from people and want to know if they recognize a virus. And what we're doing is a plaque assay, basically, right? We have here about 10 or 11 plaque-forming units of virus, and we mix this with different dilutions of antibodies. And so we have on the top here, let's start with the bottom. The bottom is our control. We're diluting the antibody in phosphate buffered saline. So here we're just using dilutions of PBS, and no matter how much PBS you put in, you still get 11 plaques. Because there's no antibody to neutralize infectivity. On the top left is a 1 to 10 dilution of your antiserum. So we take blood from you, we separate out the cells, and what the clear part that's left, we dilute it 1 to 10. We mix it with virus before infecting the cells, and you can see there are no PFU. 1 to 100, we have no PFU. But now if we dilute it 1 to 1,000, we have a little bit of virus infection, 2 PFU. And 1 to 10,000, now there's no more neutralizing activity. So that's how we detect neutralizing antibodies. It could be in people or it could be in experimental animals. By blocking infection, if you're lucky enough that your virus forms plaques, you can do a plaque assay. You could obviously do any other number of virus infectivity assays as well. So often when people are trying to be diagnosed for a virus infection, you can take serum at different times after infection and ask whether it has neutralizing activity. It's a good complement to finding the virus there as well. In fact, if you can show in a person you have virus by, say, PCR, nucleic acid, and antibodies by this kind of assay, that's really good evidence that an infection has occurred. So that is what a neutralizing antibody is, all right? It blocks infection. Now, not all antibodies we make against the virus will block infection, because we make antibodies against all different parts of viruses, but the ones that do block are called neutralizing antibodies. So here's an experiment where an animal was infected with poliovirus, and then at different days after immunization, serum was taken and antibodies were measured. And here we're looking at the antibody titer, and this is simply the reciprocal of the dilution of the antibody that we saw in the previous assay. So 1 to 10, 1 to 100, 1 to 1,000 or so. So the high numbers has a lot of antibody present. And we're actually looking at the different kinds of antibody present, IgG, IgM, and IgA. Now typically, in most virus infections, IgM antibodies, the dark blue, are the first ones to be produced. They come up very quickly and then they peak. IgM typically has low affinity. So early on in an infection, I don't know if you know how antibodies are formed, but they're formed in the lymph node by repeated selection against antigen. So the genes encoding the antibodies are recombined, then hypermutated until you get a very high affinity antibody. And the IgM are the early products of that. They typically have low affinity, but 5 IgMs can bind antigen. They're all linked at their Fc portion, so they have high avidity to make up for that. And then later on, the IgGs come up, which are the higher affinity antibodies, and then you can see IgAs are a little bit slower. But again, these persist for a long time after immunization. Eventually, the levels will go down. So the levels of antibody in your blood will all go down to very low levels. You have a little bit, but you don't need to have a lot, because if you get infected again, you're going to have a memory response which will produce a lot of antibody immediately. This is a typical time course of antibody production. Now, these are days after immunization, and in a polio infection, the infection would largely be done by day 10 or 15. So the antibodies are not really present early enough to do anything. It's really the CTLs that are curing infection. Now, I told you earlier that antibodies are important for protection, and this is an experiment that shows that. Of course, we know that immunizing people or animals, protects them against virus infection. But here's an experiment that does that. This is an animal which is being infected with poliovirus, and the outcome is paralysis. So on the y-axis, we're looking at the percent animals that are paralyzed. And what we're doing is giving the animals antibody by injection. This is called passive immunization. So we've immunized another animal against poliovirus and taken their serum, and now we are injecting it into these animals before infecting them with polio. And we inject different amounts of antibody into these animals. From none, the control animals get no antibody, and then these get progressively more antibodies. So the control animals are all paralyzed, 100%. They got no antibody. Those animals that received a low amount of antibody, again, they're mostly paralyzed. You put twice as much antibody in, now you have 60% paralysis. Here at number 11, you have less than 20% paralysis, and then when the titer is 22, you have no paralysis. So those animals are protected against polio paralysis because the antibody is in the blood. When you inject virus, the antibody combines with it and prevents it from infecting cells. So that's called passive immunization because those antibodies only last maybe a month at the most. Today we know how to engineer antibodies so that they have longer half-lives in the blood, but this is only a stopgap measure for protecting people. You may remember during the Ebola outbreak that some of the recovered people early on, we were taking their serum and injecting them into other people to try and cure their infection. And this is the basis for that, neutralizing the virus. That previous experiment was done by taking serum from an immunized animal and injecting it into another animal. But antibody can also be present at mucosal surfaces, and that's illustrated here. Here's a mucosal epithelial cell, the apical surface up at the top here. This could be the gut or the respiratory tract, the lamina propria, the basolateral domain here. And just beneath the basement membrane of these epithelial sheets are plasma cells. So these would have come here from a neighboring lymph node. So if there was an infection in this mucosal epithelial cell, the dendritic cell would have sampled the area, gone back to the lymph node, and then produce, B cell would recognize the antigen. You'd have the production of plasma cells which make antibodies. And these plasma cells home to the mucosal epithelium, and they produce IgA. So IgA, immunoglobulin type A, is the antibody which is going to be secreted. And that's taken up at the basolateral domain by endocytosis. It moves across the cell. So this is basically an endocytic pathway moving contents from the basolateral domain to the apical domain. So that's called transcytosis. And eventually at the apical domain, these vesicles will fuse, release the antibodies. They have proteins associated with them that help them be transported. Those are then removed. And then what you would have left are the red molecules, which are the antibody molecules. Right? So those antibodies would then bind viruses that are at the mucosal surface. Now, the thing about secretory antibody, or IgA, it doesn't last very long at mucosal surfaces, even less than serum antibody. So this is why we often get infected with certain respiratory pathogens. For example, the rotaviruses that cause gastroenteritis. The prime determinant of protection in your gut is secretory IgA, but after an infection, this declines very rapidly, and so you're not protected again. You get reinfected over and over. Even though you have high serum antibodies, it doesn't help to protect your gut. Antibodies, we've talked very broadly about how they work. I tell you they neutralize viral infectivity. They actually do much more than that, but let's first start with explaining how neutralization works. So here's a virus, happens to be rhinovirus, attaching to cell surface receptors and being taken up into the cell by endocytosis, and the RNA is released. This is a process we've talked about before. If we introduce antibodies, neutralizing antibodies, they can interfere at several steps in this process. So here on the right panel, we have a rhinovirus being bound by antibodies. So these antibodies are binding to sequences on the surface of the virus particle. And that binding can have several effects. It can block attachment. In fact, many people just intuitively think that this is how antibodies must work, by blocking virus attachment to cells. And certainly a subfraction of antibodies do that, but by no means is it the major mechanism. It's just, for us, makes a lot of sense to think of antibodies blocking attachment because they're blocking the receptor attachment site. But anyway, some of them do, but many of them block in other ways. Some antibodies cause the viruses to aggregate. So there's such big masses of viruses and antibodies, they can't effectively infect the cell. Some antibodies can block endocytosis, and some antibodies can block uncoating. So the antibodies can be taken up into the endosome with the virus particle, remain attached to it, and then uncoating, the release of RNA is inhibited. So for many viruses, the antibody seems to fix the virus in a state that, for example, doesn't respond to low pH. So there's no low pH mediated fusion when the antibody is present, so the genome can't get out. So the result is the virus remains in the endosome, and the next step, of course, is the lysosome will fuse with this, and then it will degrade the virus particle. So normally viruses want to get out before they encounter the lysosome. So those are three ways that antiviral antibodies can neutralize virus infectivity by binding directly to the virus particle. And here's a really nice picture of a virus bound to neutralizing antibodies. This is poliovirus, and so the virus is in different colors, the sphere. The antibodies are in purple or magenta, I guess. It's magenta. And they're binding, so these are Y-shaped molecules. You can't really see that very well, but they're binding to the virus particle. You can see these are binding at the 5-fold axes of symmetry. So there's a 5-fold axis. Remember, 1, 2, 3, 4, 5 copies of the virus subunit around it. This doesn't always have to be the case. It depends where the epitope is that the antibody is recognizing. In this case, it happens to be at the 5-fold axis. And since it's repeated 5 times around each 5-fold axis, you get 1, 2, 3, 4, 5 antibodies at each 5-fold axis. And how many 5-fold axes are there? 12. So you're going to get 60 antibodies bound, saturating to the virus particle. We actually don't think we need that many to be binding to neutralize virus infectivity. Probably just a few. In fact, for some viruses, it's been shown you just need a few antibodies to neutralize. But this is what it would look like. Of course, antibodies can bind other places on the capsid as well and have different effects depending on where they're binding. So those are examples of how antibody binding to a virus will block infection. And that's summarized at the top here. Here's the schematic of a virus. We have a neutralizing antibody, which is protecting the cell from infection. There are other ways these antibodies can work, and I want to tell you about those. One is shown here as complement-mediated lysis and phagocytosis. So if an antibody binds an enveloped virus to a glycoprotein, right? Complement, which is the collection of serum proteins that works together with antibodies, that will recognize the antibody and will lyse the viral membrane. All right, so complement recognizes the antibody bound to the virus particle, and it recognizes it as foreign because it's able to do that, can lyse the virus particle. The other thing it can do, if we have a virus particle with antibodies bound to it, the Fc portions are sticking out. Cells with Fc receptors can bind those antibodies and take them up. Like macrophages have Fc receptors, they will bind this and phagocytose it. That's called opsonization, and phagocytosis into the macrophage will end up destroying the virus particle. And on the bottom are all the different things that can happen to a virus-infected cell by antibodies. So we've just focused on antibodies binding to viruses, but they can also bind to virus-infected cells. And in cells infected with many different viruses, there are viral antigens on the surface, right? The glycoproteins are inserted initially in the plasma membrane. Those can be recognized by antibodies. So this doesn't work for icosahedral viruses that are non-enveloped because there's nothing on the surface. But for viruses that put viral glycoproteins in the plasma membrane, the antibody can recognize those. The binding of antibody can have a number of consequences, which are shown here. The cell can be lysed by complement, and that's infected cell lysis or clearance. There's some evidence that binding of the antibody to a glycoprotein can cause signal transduction into the cell, which will inhibit virus replication. These antibodies might inhibit virus release. Remember, these viral glycoproteins are going to eventually be the basis of a new virus particle formed by budding, so the antibody can prevent that process. And the antibodies can also prevent cell-to-cell transmission. In some cases, viruses go from one cell to another by fusion, and the antibodies can prevent that as well. So while neutralization of virus particles is important, don't forget that antibodies against viral proteins can also operate in the infected cell. Do you think viruses have a way to get around antibodies? Yeah, I mean, you have to say yes, right? Whenever I ask you that question, the answer is yes. Absolutely. Here are two examples. On the left is rhinovirus, one of the viruses that causes the common cold, an upper respiratory tract infection. And this is very related to poliovirus, so antibodies bound to rhinovirus would look a lot like the picture I just showed you a few slides ago. What rhinovirus does is that there are over 150 different genotypes of the virus, and antibodies against one do not block infection with another one. So that's the strategy of this virus, to make a lot of different genotypes. So in your lifetime, you could get 2 to 3 colds a year by different genotypes, and that would last you, right? If you live to 50, that would get you through 150 genotypes. And these genotypes are globally distributed. So the strategy is to make lots of different antigenically unrelated viruses. All right, so that's one strategy by rhino. The other is exemplified by influenza virus. And on the right is the hemagglutinin of influenza virus. Remember, this is one of the 2 major glycoproteins in the viral envelope. The hemagglutinin is responsible for attaching to cell receptors and catalyzing fusion in the endosome. So antibodies against flu that neutralize infectivity, many of them are directed against the HA. They can block attachment, they can block endocytosis, they can block fusion. What influenza virus does is to vary the sequence of the hemagglutinin protein. And on this model of the hemagglutinin, the HA, this is a trimer, remember. There is the globular head, which has the receptor binding site, and this part at the bottom would be attached to the virus particle. These different colored sites change in influenza viruses from year to year. And they also happen to be sites where antibodies bind. So there are lots of antibody bonding sites on the globular head. You could see yellow, orange, green, blue, magenta. And so let's say you get a flu infection one year, you make some antibodies against these, and then the next year the flu strain that's arisen has one amino acid change in one of these sites, and that's all it takes to block the antibody from binding. So antigenic variation is the way influenza gets around antibody responses, and that's one reason why you may have to get a flu vaccine every year, because the virus is always changing. It's one of the few viruses that we know that does this. HIV does this in an infected person. HIV changes in you over the years that you were infected. You make antibodies against it and the virus changes. You make new antibodies, the virus changes again. So flu and HIV are very similar in that sense. Now, down here in the parts of the HA that are closer to the membrane, the blue and the red epitopes, these are in a very different part of the protein. These are in the stalk, not in the globular head. And these areas, while antibodies against these areas will neutralize infection, these areas don't change. They're highly conserved because apparently the structure of the stalk is very important and you can't mess with it, whereas the head can be changed. So people are now trying to make vaccines which give you the stalk part and which will give you antibodies which should protect you for your lifetime. So maybe one flu shot when you're a kid, and that takes care of you for 10 or 15 years. That would be nice. So we'll talk more about that separately. So those are just 2 ways that viruses get around antibodies, just like they can get around everything else. The next question, which statement about antiviral antibodies is incorrect? 1, they are important for protection against viral infections. 2, they only neutralize virus infectivity. 3, they may block virus attachment to cells. 4, they can be found at mucosal surfaces. And 5, IgM is the first to appear, then IgG. So which is wrong? 89% of you picked B. They only neutralize virus infectivity, and that's not right. Yes, antibodies do more than that. They can neutralize virus infectivity. They can also do things to the infected cell that prevents virus production, for example. Some of you picked E. IgM is the first to appear. That's correct. All right, so that's antibodies. We'll come back to antibodies in a moment. The other arm of the adaptive response is cell-mediated immunity. These are CD8 T cells that have become cytotoxic T cells. And these are important or essential for clearing most virus infections. Not antibody, but cell-mediated immunity. So again, the dendritic cell has gone in the lymph node. It's presented antigen to a CD8 cell. It's foreign. It's detected as foreign. And that CD8 differentiates into a cytotoxic T lymphocyte. And one of the features of a cytotoxic T lymphocyte, or CTL, is that the cytoplasm is full of granules. These granules contain proteins called perforin and granzymes. And what happens is the CTL comes out of the lymph node and it homes to the infected cells by picking up a gradient of cytokines produced by the infected cells. Those infected cells are going to be displaying viral peptides on MHC1 on their surfaces, right? Because that's what happens with the endogenous antigen presentation. And the CD8s are going to come along and look to see if there's a foreign peptide on the surface of the cell. So here on the bottom is the infected cell. MHC1 is displaying a peptide. The CTL comes over, the T cell receptor looks to see if the peptide is foreign. It should be. It's one of the ones that was presented in the lymph node. And there's also another receptor interaction that's necessary for this interaction to occur. And if this CTL finds that this is a virus-infected cell, it will deliver granules containing perforin and granzyme to the cell, and they will induce apoptosis. All right, so we used to think that it actually poked holes in the infected cell, but it turns out that the packets that the CTL deliver to the infected cell induce apoptosis, so this infected cell dies. So you can see the idea here is to stop virus replication, because the infected cell, a dead cell, can't produce viruses, right? So the main killing is by apoptosis. Of course, there are countermeasures at every step of this whole process. One of the perhaps more interesting countermeasures is that viruses can change those peptide sequences. Just like influenza can change the HA so it doesn't bind antibody anymore, viruses can change the sequence of the peptide recognized by CTLs. So now, for example, the infected cell will put up a peptide that's not recognized by the CTL, will go away and it won't lyse it. And this happens in HIV-infected individuals over time. They generate what we call CD8 peptide variants. So again, without countermeasures, viruses would be eliminated by this wonderful immune system. So this is an experiment, again, in an animal, which gives you a sense for the kinetics of production of these CD8 positive cells. So this is an animal infected with a virus. We're looking at days after infection. There are two panels. On the top, we're looking at virus production in the spleen of these animals. This virus targets the spleen. So this is PFU per spleen, the total PFU per spleen. So you can see the virus titer after inoculation at day 0 peaks very quickly within a few days in the spleen and then goes down. And on the bottom is a graph where they have taken lymphocytes out of this animal and asked what fraction of them will kill cells, percent lysis, and those are effector CD8s. Those are the green bars. So in other words, you remove CD8 positive lymphocytes from the animal, and then you add them to infected cells in culture and ask, can they lyse those infected cells by virtue of being CTLs? And so you can see by day 10, you get a peak of effector CD8 CTLs. Again, effector meaning they're killing the cells. And so they come up just after virus titer is beginning to come down. So it doesn't really tell you cause and effect here. And eventually those are eventually regulated. You want to get rid of those CTLs because you don't want them there when they're no longer needed. However, if you just look for CD8 lymphocytes that are epitope specific, in other words, that recognize a viral epitope, those go up and persist for a long time. So those cells are going to be with you for ages and will become CTLs again if you are reinfected. but the CTL activity is rapidly shut down. So this is a rapidly occurring event. So what's important for a virus infection? Is it antibody or cell-mediated immunity? So here's an experiment where we're simply asking, which of the two do you need to protect against monkeypox infection of monkeys? So just one particular virus. You could do this with different viruses and it would be different. So what we do here is we immunize the monkeys with an activated virus at day 0, and then we do some sort of immune manipulation. After a while, we measure the antibodies present by it's day 22. So we're asking, did the vaccination result in any antibody production? Then we infect them with virus at day 28, and then we see if they die. Fatality is the endpoint of this infection. So here we have monkeys that are vaccinated. We don't do anything to manipulate their immune system. On day 22, they have high titers of neutralizing antibodies. These are the reciprocals of the dilutions at which you get inhibition of virus infection. You infect these animals, and they're all protected. No fatalities. Now, it's only 4 because these are monkeys. You can't do 100 monkeys when you would have no money left. All right? So any monkey experiment is done with few animals. But they're all protected. And that is presumably because they have antibodies. But let's do an experiment to figure that out. The next set of monkeys, you immunize them, and then you deplete their B cells. You can do this in a variety of ways. And then you see by day 22, they don't make much antibody, which makes sense because you've taken away their B cells. And then you infect them, and 3 out of 4 of them die. So this would tell us that antibodies are really important for protection against infection. You take away their CD8 cells, which you can do, that doesn't affect antibody production. It should not. Right? You infect them, they're all dying. So CD8s don't protect you against infection in this experiment. It's the antibodies that do. So really, depending on the virus, one or the other response may be more important for either protection or recovery. CTL versus antibody response. And it varies according to the virus. As you've seen, the protection against smallpox needs antibodies, not CD8s, but it's different for other viruses. So how is this determined, whether an immune response will make antibodies or CTLs, or what mix of both is it going to make? And the decision is made very early on when the dendritic cells go into the lymph node and interact with B and T cells. So remember, they're picking up antigens from the infected cells. They're going in the lymph node. They're presenting the antigens. They're also making cytokines. And apparently the nature of the antigen, the virus that's there, can influence the cytokines that are produced. And this, in turn, can tip the balance between antibody production and CTL production. So that happens in the lymph node. So in the lymph node, remember, the dendritic cells contact the CD8 and the CD4 cells. And the information that they exchange there, particularly with the CD4 cells, determines whether they make one kind of cytokine or the other. So remember, Th1 cytokines favor cell-mediated immunity. They favor the production of CTLs. The production of Th2 cytokines favors the production of antibodies. So when that DC goes into the lymph node and interacts with the CD4 cell, depending on what it gives that cell, it will bias the CD4 cell to make either Th2 or Th1 antibodies. Now, it's usually not all or none. It's usually biased in one direction or another, depending on the virus. Some viruses, antibody responses dominate. Some cell-mediated immunity dominates. But the interesting thing is that there's cross-regulation. Let's say the interaction of the dendritic cell has biased the CD4s to make Th2 cytokines. So these include things like IL-4, 5, and 10. Those cytokines suppress the production of Th1 cytokines, and that's shown down here. And likewise, if you make Th1 cytokines, those suppress Th2. So whatever dominates initially continues to dominate because of this crosstalk. So the kinds of cytokines that are produced by the CD4 cells in response to the dendritic cells determine whether that infection is going to be responded to with antibodies or CTLs. And when we talk about vaccination, we'll see that this makes a big difference. And your vaccine has to do the right thing. If your vaccine induces the wrong kind of Th response, it's not going to work. And we have wonderful examples, well, not so wonderful. We have examples of how that has gone awry, which we'll talk about. All right, our next question. For some infections, CTLs are more important for protection than antibody. How is the CTL antibody balance determined? 1, by toll-like receptors, 2, by intrinsic defenses, 3, by autophagy of infected cells, 4, by the mix of peptides and cytokines presented by DCs, 5, it depends on whether the capsid is icosahedral or helical. All right, let's take a look. 96% of you picked D, by the mix of peptides and cytokines presented by DCs. So a couple of you picked C, by autophagy of infected cells. So that certainly is upstream, right? The DC is picking up pieces of dead and dying cells. But the real determinant is what peptides and cytokines are presented to the dendritic cells, to the lymph node cells. Now, adaptive responses have memory, unlike intrinsic and innate responses. So if you get infected after you've survived your first infection, you respond very rapidly and very specifically to that original pathogen. And again, innate and intrinsic responses don't have memory. And this is why we vaccinate, because when you vaccinate, you give someone a virus without giving them disease, and they develop memory, so they will be protected next time. So here's a natural experiment done many years ago, which demonstrates immune memory. This was done on the Faroe Islands, which are here. This is the United Kingdom and Europe. Those are the Faroe Islands. So at one time in history, you couldn't get there very easily. You had to take a boat, and there weren't any boats that went to these islands. In 1781, this was studied by an epidemiologist. There was an outbreak of measles on these islands. For the next 65 years, there was no measles on the island because everyone was immune. But of course, during that time, new kids were born who didn't have immunity. And then in 1846, apparently a ship came with some measles-infected people on it. There was another outbreak of measles, but nobody who had survived the original outbreak were infected. So there were some people around from that original outbreak. They were protected because they have long-term memory. So memory lasts a long time, and you don't need virus around to maintain it. It's something that you maintain on your own. You maintain cells that are specific for the pathogen. So how does that work? So you get, here's a graph of time versus antibody or T cell production. So memory applies to both antibody and T cell production. We have a first infection here, which eventually gives you a response. It could be antibody, it could be CTL response. And then that response eventually wanes. Remember, I told you your antibodies and CTLs don't last forever. And in fact, I showed you evidence for that. And then maybe many years later, you have another infection with the same pathogen. You have a very, very rapid inducing of antibodies or T cells. So you don't have this 14 to 21 day delay. And that protects you from infection. You either get a mild infection or no infection at all. So that's why memory is valuable. Memory consists of cells that persist in us for a long time. And it happens at both the B and T cell level. So for B cells, there are two kinds of memory that we recognize. We recognize that there are memory B cells, such as this one. So this is a B cell that has a specific antibody on its surface that's recognizing the virus that infected you. These persist in the spleen and lymph nodes for years. And when you are infected again, they will be there to respond very quickly. So the antibody rearrangement and hypermutation doesn't have to occur. It's already, all has to occur is these cells have to divide, so you immediately make high affinity antibody in high quantities. So that's why it's good to have those cells around. We also, and these don't make antibodies unless antigen comes along. So they're not just sitting there making antibody randomly, they just have it membrane bound. We also know that there are long-lived plasma cells. So these are B cells that have differentiated and can produce high quantities of antibody. These tend to be in the bone marrow. So they can come out, again, when you are infected. How would they know to come out? Well, your infected cells would make cytokines that would be detected by the plasma cell, and then they would come out and make antibodies. We also know that there are memory T cells that last a long time in us and can they are specific for an epitope that's been delivered to them by the dendritic cell. They last a long time, and they can be reactivated at any time. And this is one of the reservoirs of HIV, and why it's really hard to get rid of, because these are very, very long-lasting cells. They may last your lifetime, as far as we can tell. So here's some numbers to give you an idea of how long-lasting immunity can be. This is actually just the persistence of antibody. Very low levels of antibody can persist for a long time. So these are viruses that cause systemic infections, chikungunya, measles, mumps, polio, etc. Look at many, many years, 30 years, 65 years, 75 years. That's the longest against vaccinia virus. So this is antibody in your blood, not being made by memory cells. We know there are memory cells present, but also you have a low level of antibody. But what's interesting is that for viruses that cause mucosal infection, the antibody is less durable. You can see in terms of months, not years, for these viruses that cause mucosal infection. So this is serum antibody, right? Not secretory. Secretory is gone very rapidly. But even serum antibody is not durable. What are the following characterizes immune memory? Memory B cells, long-lived plasma cells, lasting a long time without more exposure to virus, humoral antibodies that last a long time, all of the above. 89% of you said all of the above, which is right. All of these things I just mentioned, they are all characteristic of immune memory. All right, the last topic I want to talk about is the damage that the immune response can cause. We already talked last time about how interferons cause flu-like symptoms, right? Well, antibodies and CTLs do even more damage. We call this immunopathology. And many of the clinical symptoms of a viral disease, we've talked about fever, nausea, laziness, and so forth. But tissue damage, pains, these are all caused by the antibody and CTL, the cell-mediated response. And in fact, there are many viruses that don't kill cells. They don't cause cytopathic effects, yet they do cause tissue damage in an animal. That tissue damage is largely caused by the immune response. So here's an example of different ways that immune responses can cause damage. This is immunopathology. There are mechanisms caused by CD8 T cells, by CD4 T cells of both Th1 and Th2 classes, different cytokine secretions. There's also pathology caused by B cells. So let's look at a couple of these to see how they work. This is an example of a virus, lymphocytic choriomeningitis virus, which is a rodent virus that's used extensively to study immunology. You affect mice with LCMV, they're dead within 8 days. If you immune suppress the mice with steroids to prevent the development of an immune response, the mice live. They become persistently infected. But if you inject them with CD8 positive T cells that you've taken from another immunized animal, it kills them. So it's the CD8 positive T cells that are causing the mice to die. They're causing tissue damage. So here is one of the reasons why this virus does that. On the top two panels are infections of mice with LCMV. On the left, we're looking simply at the percent that are alive, 100% alive. So wild-type mice, the plus, these mice are dead within 10 days after infection. But if you knock out the gene for perforin, perforin is a protein in CTLs that's responsible in part for inducing apoptosis. If you knock out the gene for perforin, these mice survive infection, or the light green lines there. So perforin in the CTL is causing them to die. On the right is a measurement of liver enzyme in the serum of these animals. So when the virus is damaging liver, the cells release enzymes, which you can easily assay. So this is one particular enzyme of the liver. And you can see wild-type mice plus, the dark green, the serum enzyme is rising. If you knock out perforin, you don't get any liver enzyme in the serum. The CTLs are not damaging liver cells, so the release of enzymes into the circulation is a cause of CTLs lysing the infected cells. So that's an example of CD8 positive damage. Now on the bottom is a different virus, it's a Coxsackie virus, which is related to polio. And in humans, you can have Coxsackie infections of the heart. You don't know it until it's too late, and then you need a heart transplant. So they're a major indication for heart transplantation. Turns out the damage they cause to the heart is entirely immunopathological. This is a mouse infected with a Coxsackie virus. It's a section of heart tissue, and it's stained with a dye that stains light blue, and this shows you the calcification caused by virus infection. So this is pathology. On the right is a mouse lacking the gene for perforin. And so they're virus infected, but the heart tissue is absolutely normal, and these mice survive. So this is very important to understand that the immune response can do bad things. CD4 positive T cells can also cause pathology. These cells, as we've talked about, make a lot of cytokines, either Th1 or Th2 type cytokines. They recruit cells like neutrophils and mononuclear cells that can cause damage. And they release proteases. These cells release proteases, reactive radicals, and cytokines like TNF-alpha that also cause cell damage. So CD4 cells, they may seem innocent because they don't lyse cells, but they can produce proteins that are damaging. And so there is a disease of humans called herpes stromal keratitis, a very common cause of blindness in developed countries. It's entirely immunopathological, and it's caused by CD4 positive Th1 cells coming into the eye. And this can cause blindness. You can get repeated infections because you're not immune, and eventually the eye becomes opaque, as shown here, and you can't see. Now, the reason this happens is the virus infects the corneal epithelium. So these are two sections of the cornea at different magnifications. The epithelial cell layer is at the top, staining darkly. The virus infects these cells, but there's a CD4 response to infection, CD4 Th1 in particular, and these cells secrete cytokines and damage the stromal cells, which is the underlying cell layer on either side. And on the magnification at right, you can see lots of dark cells here, and those are CD4 Th1 cells. You can tell by staining them that are coming in. They're causing damage to the stroma, and that's what makes it opaque and causes a blindness. So the virus is not in the stroma. It's in the epithelial cell layer, but that infection is attracting CD4s to the stroma, and their secretion of cytokines causes problems. So obviously this is important to know because maybe we can determine ways to intervene pharmacologically to prevent the damage if we knew, for example, which cytokines were doing that. This is an example of how tumor necrosis factor can cause encephalitis. This is a mouse model for West Nile virus encephalitis. When you infect mice peripherally with the virus, it gets into the brain and it causes brain permeability. So you can see that here on this top level of brains taken from these animals. So these are animals infected, these are wild type animals infected with virus, and they're given a dye that if the brain barrier has become permeable will stain the brain blue. So you can see by day 3 the brain has become permeable. If you knock out toll-like receptor 3 gene, you delay the permeabilization of the brain after virus infection. And so what's happening is TLR3 is sensing the virus, and one of the cytokines produced is TNF-alpha, which compromises the blood-brain barrier. So if you take away the sensing of infection, you delay the compromising of that. You delay the permeabilization of the blood-brain barrier, and the virus, the TNF doesn't respond. So this is an example of cytokine-mediated immunopathology. We don't know if this happens in humans. We can't do this experiment. But the lesson is clear that these compounds, which can have uses to destroy infected cells, can also lead to virus invasion of the brain. So I should add that if you infect toll-like receptor negative mice with the virus, not only does the dye not get into the brain, but the virus doesn't as well, so you don't have encephalitis. So the implication is that the TNF is permeabilizing the brain. This is a peripheral response to infection and allows the virus to get in. The poxes and rashes that we see as a consequence of many virus infections are the result of Th1 cells that home to the skin. They make cytokines. Those cytokines increase capillary permeability. It's one of the things that they do. It attracts more T cells to the area, and you get the little rashes that are so typical. So this is a form of immunopathology. Dengue fever is a great example of B-cell mediated immunopathology mediated by antibodies. This is a mosquito-borne virus that is endemic in many parts of the world. There are about 50 million infections a year. The primary infection is transmitted by these mosquitoes, which are globally distributed. Here in yellow is where Aedes aegypti are present. This virus this mosquito was not in South America prior to 1981, but came back as a result of the tire trade. This is a theme that we've visited before when we talked about chikungunya. So Aedes aegypti spreads the infection. The primary infection is not too serious. Febrile illness with aches and pains in the bones, usually recover. Sometimes you get a more serious disease, which is accompanied by hemorrhage and loss of fluid. This happens in 1 in 14,000 primary infections. You make antibodies against the virus, but the problem is there are 4 serotypes. So if you're infected with, say, serotype 1 of dengue, and then you get infected another time with serotype 2, you get a more serious disease because the antibodies against the memory response against that first infection will bind serotype 2, say, but they won't neutralize its infectivity. And what's worse, they'll allow it to get into cells where they don't normally infect. So here we have antibody binding a dengue virus. If this were type 1 antibody against type 1 dengue, it would neutralize infection. But if you get infected with type 2, you'll make type 1 memory antibodies. They'll bind type 2 virus, but they won't block infection, but they will allow the virus to get into, say, macrophages via the Fc receptor. Macrophages have Fc receptors. The antibody will bind it. The virus will get into a macrophage where it doesn't belong. The macrophage then produces all kinds of cytokines, which are diagrammed here, attracts CD4 and CD8 cells. You get capillary permeability. So you have a very serious disease. And the incidence after secondary infections is 1 in 90 and 1 in 50 compared to 1 in 14,000. So this is why dengue is serious, because the first infection is okay, but it's that second infection that can kill you. And so right now we're trying to make a vaccine that would protect everyone in these areas against all 4 serotypes.